Hello, the title of the work that I'm going to present is Subcortical Anatomy of the Ventral and Dorsal Attention Networks. Attention is a core cognitive function that undertakes the filtering and selection of relevant information by the brain. Two macroscale networks regulate attention mechanisms. The Ventral Attention Network, in orange, is a stimulus-driven system, while the Dorsal Attention Network, in blue, mediates goal-driven tasks. The neuroanatomical study of these networks has taken a predominantly corticocentric approach. However, some evidence suggests that they may also be composed by subcortical structures. First, the electrophysiological recordings in primates has revealed that subcortical structures such as the pulvinar and the superior colliculi also regulate attention. In addition, the analysis of distant phylogenetic species with markedly different cortical morphologies, such as pigeons, has shown that they share behavioral similarities with us during attention tasks. Finally, in humans, neglect, which is a paradigmatic syndrome of hemispatial inattention, does not only arise after cortical dysfunction, but also after subcortical lesions. The alignment of individual functional maps exclusively based on structural features may lead to a misalignment of small subcortical structures. In a previous work, we have shown that if we do an additional step of functional alignment, we may improve the identification of these small structures in average group analysis. Therefore, the aim of this work was to explore the subcortical anatomy of the ventral and dorsal attention networks by the functional alignment of resting state fMRI maps. Our hypothesis was that some subcortical nuclei, such as the pulvinar, the striatum, the superior colliculi, and locus coeruleus, would also integrate attention networks. To achieve this, first we computed Van and Dan maps by a seeding analysis of 110 7 Tesla resting state fMRIs from the Human Connectome project. Seeding was based on Gordon's et al. Cortical parcellation, which is composed of 23 Van cortical parcels and 32 Van parcels. Then, we performed the functional alignment of these maps by an iterative diffeomorphic transformation using hands. Group effects were analyzed with one sample permutation test. Third, we conducted a connectivity analysis. For structural connectivity, we performed a whole-brain deterministic tractography. For functional connectivity, we calculated partial correlation, and for graph theory analysis, we computed the properties of each network node. Finally, we studied the relationship of these models with the neurotransmitter system by calculating the spatial correlation of the projections of the identified brainstem nuclei with the distribution of 9 neurotransmitter systems provided by Hansen and colleagues. Here are the first results of our analysis. Namely, you can see the surface, cortical, and cerebral projections of Van and Dan maps, in which we see the typical distribution of these networks along the cortical surface of both hemispheres, along with their different projection in cerebellar lobes. When we analyze the subcortical distribution of these maps, we can clearly see some of the hypothesized subcortical nuclei, such as the pulvinar, the superior colliculi, and also a cluster of brainstem nuclei, such as the pedunculopontine system and the RAF nuclei. Actually, the conjunction analysis showed that most of these nuclei are shared by both networks. In the connectivity analysis of VAN, the structural map does not only reveal the typical association fibers, such as the third branch of the superior longitudinal fasciculus and the uncinate, but also a group of projection fibers, namely frontopulvinar and tectopulvinar. In DAN, we saw the same pattern. So this structural map does not only show the typical association fibers, in this case, the first branch of the superior longitudinal fasciculus, but also subcortical projections fibers, namely the frontopulvinar, parietopulvinar, and also tectopulvinar fibers. In addition, the graph analysis revealed that these subcortical nuclei have high values in terms of betweenness centrality and degree centrality, showing that these nuclei don't seem only to belong to these networks, but also they seem to play a core role in their organization. Finally, the correlation analysis with neurotransmitter system has shown that the distribution of VAN and DAN was specially correlated with the distribution of alpha-4 beta-2 acetylcholine nicotinic receptors, and also with the distribution of dopamine and serotonin transporters. With these results, we propose an improved model of the ventral and dorsal attention network that includes the pulvinar, the superior colliculi, the caudate, and the cluster of brainstem nuclei, whose projections are specially correlated with the distribution of acetylcholine alpha-4 beta-2 nicotinic receptors and with the dopamine and serotonin transporters. The main limitation of our work is that, as it is based on resting state analysis, we are unable to untangle the dynamic interactions that occur between these two networks. However, future investigations with task-related fMRI in humans or direct electrical recordings in animals may contribute to answer this question. Finally, we believe that the correlation that we found between the projections of the identified subcortical nuclei with the neurotransmitter system that I referred increased the plausibility of our model, since both alpha-4 beta-2 nicotine agonists and methylphenidate and modafinil, which are important modulators of dopamine reuptake, have clear and established rules in the modulation of attention. With this, we believe that our model may reconcile behavioral data from distant phylogenetic species and may also contribute to a better understanding of attentional disorders in humans. Thank you very much.